Oh, thank you. So today, I thought we will go through some basic classification of life, and it might be very basic for a lot of people. So I apologize if you know way more than what I'm going to show you. But the idea here is to make sure that we are kind of in the same page behind the idea of doing this project and some of the basics. It's hard to know, the kind of background everybody has, but we usually keep a very basic slide to get an idea of everything. Okay. So, Iqbal, could you also keep an eye on the questions, please, in case I miss it? I might not see it, so just stop me. Also, you all can interrupt me anytime. I usually have a lot of fun chatting with people, so please feel free to ask anything or jump in if you have more information. Okay, cool. So, one way to sort of classify life was given by Dr. Robert Whittaker in 1960s. And as we go through, we will talk about how there are different ways we can classify living organisms or living things. But the way Robert Whittaker did in 1960s was these different classes, and one was the huge animal class, where of course we are as mammals, but also a lot of crustaceans and insects come into that category. The other one was the large plant class, of course, which we see around all the plants, the ferns, the angiosperms. And the third class, which he gave, which became a very important one, was also the fungi. So all the mushrooms we eat and some other kinds of fungi were in that group. There was another class, which is called as Monera, which we will also talk a little bit about because it gets a little confusing with the new taxonomy classes about Monera and Protista, because what Whittaker did at that time, he basically put everything which he couldn't sort of classify at that time into the Protista group. So if you see the green algae, the brown algae were put together with like protozoans in the Protista group. But we will learn more and more about how algae can be of archaic kind or the old kind, but at the same time, it can be eukaryotes as well. In Monera, he basically put stuff like bacteria or any of those archaic microorganisms. So this was the sort of classification that was given in 1960s. But before we go into too much detail of classification, something important to keep in mind is also the length scale we have been talking about. So when we talk about length scale, it's about how big an organism is. So we know that most of the organisms and stuff is made up of, atoms, which is one of the smallest unit, which essentially can also make a lot of different stuff like DNA or proteins together. And that assembly keeps growing on, and we can have more and more complex organisms. So we know that humans, which we can see by our naked eye, or anything which is sort of visible to us, is in this range, which is usually in millimeters or meters, which we can really see with our naked eye. However, there's a whole range of stuff which is very hard for our eyes to resolve, and which is often either the microorganisms or something which we call as sort of an unresolved or super resolution state. So these are the ones which usually are somewhere around 1/1,000,000th of a meter range. So that's the micron range. And usually we need some special tools to sort of visualize this range of either the organism or a single cell. So like we can see our body, we can see our skin, but it's very hard for us to resolve and see a single cell out of it unless we use something to sort of resolve it and make it bigger for our eyes to see it, right? So we see our blood, we see like a huge red thing, but if you think about blood is also made up of these really smaller cells, which is the human red blood cells, and it's like around 8 microns. So just to give you an idea of the sort of the length scales we are dealing with. So just to give you an idea, most of the algae can also be in this range, but there are some which could be also in this range, which you can kind of see with the naked eye. But if you want to have more details at the cellular level, you have to be in this range. So you need an instrument to sort of resolve it or make it bigger for your eyes to be able to see it. Okay, So just as I said, a micron is 1 millionth part of the meter. And this is sort of a fun thing you can do at home once you receive your foldscope, that you can look at a grain of salt under it versus you can also look at the bread mold. Please don't look for anthrax. I hope you don't find anthrax around you. But this is really to give a range of stuff, right? So you can look at human hair, which is around 100 microns, and bread molds or the spores from that is really, really tiny. And you will see how your foldscope is able to resolve, let's say, a hair versus a bread mold. And that would be a very interesting experiment you can do in the back of your N. I will also really suggest to look at pollen grains because they are beautiful structures. And if you look at different pollen grains from different flowers, they all have different structures. Like some are spherical, some are triangular in nature, and they're really, really beautiful to view under. And I think Iqbal in coming classes will tell you how to use a foldscope, but also give you some examples of these things, and you can do it at home as well. But again, just to give you an idea, if you look at the human hair here, which is like the 100 micrometer diameter, something like a bread mold will look that much smaller to the human hair. And here is a grain of salt there. Okay, so again, coming back to the classification of the living things, one way to classify living things can be also based on their organization. So some organisms are just unicellular. That means it's just a single cell. So here is an example of amoeba. But you can also see these beautiful algae, which are unicellular in nature. So there is one example here. So this is just one single cell. However, we know most of us are formed by multicellular, right? So we are a multicellular organism because our skin, our organs, everything is made up of multiple cells that are stacked together. So it's like bricks coming together next to each other and forming a whole organ or an organism. However, a lot of algae are also multicellular in nature. So just to give you an idea that algae is so broad in terms of their characterization and classification, because you can have a unicellular one versus you can also have a multicellular one, which are stacked to each other. And eventually, like there are algae which you can see by naked eye. And when you try to look in the cellular level, you will see that they're really this beautiful stacked system and is a multicellular organism. The other way to classify organisms is to think about their evolution. It can be a prokaryotic cell or it can be a eukaryotic cell. So one of the examples of prokaryotic cell is actually bacteria. And we are a eukaryotic organism. That means all of our cells are actually of eukaryotic origin. And one of the major differences between a prokaryote and a eukaryotic system in terms of evolution is the complexity of it. So prokaryotic cells usually have a very simplistic system. So here is an example of bacteria where it moves either with a flagella or moves with these help of these pili or ciliary-like structures. However, inside, most of its DNA material is just like lying in the protoplasm there. So it's all open system. So there is not so much of an organization going on. And there is this beautiful cell wall outside, which is made up of different sugars to encapsulate the whole thing. However, when we talk about the eukaryotic system, you can right away see how much more complex it is. But one way the eukaryotic system developed was really making these different organelles and really encapsulating that in membrane structures. So not only there is a huge membrane here, but most of the organelles inside also have membranes which keep the stuff together. So if you look at the nucleus here, all the DNA and all the genetic material in this is really inside a nucleus, which also has a nuclear envelope. That's again a membrane, which is keeping all the DNA together in one system. Whereas again, going back to prokaryotes, you can clearly see that the DNA is really circular. There is no such membrane around it and it's just lying in the protoplasm. So this is the complexity that happened during evolution. And this is really to do more and more complex functions and have a very energy conserved system that we developed into a eukaryotic cell system. So again, this is one way to think about these classifications as well. The other way to think about the classification is easily what we learn in school. The first thing, right, is the animal cell and the plant cell. And here, just to go back, this is really, we are just talking about the eukaryotic system now divided into two different kinds, which is the plant cells, which are the food making cells, and it has chloroplast, and we will talk a little bit about it. So they undergo this whole process of photosynthesis where they use energy from the sunlight or other light, and then they make food. And the whole plant cell is sort of formed in a way that it helps the process of photosynthesis. So it's full of chloroplast. So all the plants we see is green because of the green pigment chlorophyll, and we'll go in a little bit in detail about it. But the other really thing that is important for a plant cell is the cell wall, and it's majorly made up of cellulose, right? So, and the cell wall is important for a plant cell because it needs that rigidity in order to stand tall, in order to, face all the rain or the weather changes, everything that happens. So that cell wall is also something that evolved in a plant cell along with the whole chloroplast system. However, what we have is mostly mitochondria. So you see these mitochondria lying, and we have a more flexible cell membrane. So we don't have a cell wall in animal cell. And one of the reasons is that because we, if you look at just our body and think about different organs we have, like just think about your eyes versus your skin versus anything else, all of them really are made up of different kinds of animal cells. So you need these sort of, I would say, fluidity in the system to change shapes. And that's really what animal cell has evolved to do. So its cell membrane is such that it can do those cell shape changes, especially during development. So when we as humans or mammals are developing from a single ovum, it undergoes a lot of division, but then it specializes to form different things like your neurons or your skin again, or something else like a muscle. It needs to undergo those cell shape changes and specialization. So the animal cell is evolved to do that massively. So it's not its job to make food as such or whatever, right? So it's not using energy from outside, but it's actually making energy inside, which is the ATP, to sustain and to go. And that's usually because of all the mitochondria it has. Not that a plant cell doesn't have it, but its major function is via chloroplast and via photosynthesis. So again, why have we been talking about it a lot? Because again, we wanted to focus on organisms that are microscopic in nature. So when we say organism that is microscopic, that means it's hard for us to see it with our naked eyes. And that really ranges from anywhere 1 micron to 100 microns that, it's very, very hard for us to see. Anything below that, it's even harder for conventional light microscopes, which we will talk about, to resolve. So you have to use some other techniques like X-ray crystallography and stuff to really resolve it and know about its structure. So we are going to stick to kind of this range here. Okay, this is very basic. We don't have to go through. When we talk about microorganisms, you can also classify it based on beneficial versus harmful microorganism. We know that there are microorganisms or bacteria and stuff or yeast we really use in our daily lives, like for example, for making bread, or antibiotics were developed from it. However, we have tons of harmful microorganisms as well, which causes diseases, right? Like if we have strep throat, it's because of the strep bacteria. So as most of the microorganisms, you can also classify it as a beneficial one. Like we know that our gut flora and fauna, which we call usually, is very important because we keep drinking probiotics nowadays, is because to make our gut bacteria, which is the good bacteria, keep going on. At the same time, you could have an infection by a bad bacteria, so you need that kind of a good balance there. Okay, so now thinking about the microorganisms and what could really resolve that. So one of the way we can look at these microorganisms is by magnifying these tiny objects. And for that, what we need is a powerful sort of a magnifying glass. So the way microscopes were developed were really because of these optical properties, which got, sort of people found out in the early days. And we'll go a little bit in detail about it. So one of the questions we could ask always is, of course, why would one use a microscope? And I've been kind of answering that already, that if you want to really see it or resolve it, get the details out of it, you need an instrument to make it big enough, or make it seem big enough to our eyes so we can actually see it. And the second thing is, what are the basic principles on which a microscope, which is the instrument, works on, right? I might be going really fast, so please feel free to stop me. Okay, I'm going to minimize here because I can't see my own slide. Okay, so how do we magnify stuff? So I think most of you might have an idea of using a magnifying glass, because that's the basic we all have used. And it's basically just a lens, right? The property on which it really works is that the light from any object you are focusing your magnifying lens on passes this light, and that goes through the lens and it converges in your retina. So also when we are converges on your eye lens. And also one of the naturally formed beautiful lens is actually our eyes, right? So it also has this beautiful lens over here, which we know by the iris controls the amount of light that goes in. So if you look at the dark part inside your eye over here, it really controls how much light can go in versus how much we can't have it. So people like me who have glasses have issues in their lenses because of the change in the convex and concavity. So one of the things you can read more in detail about, if you already don't know about, is what is a concave lens versus what is a convex lens, kind of a homework. Okay, so one of the basics here is that light passes through is bent, and this bending of light really happens because of refraction. So what is refraction? One of the easiest way to visualize refraction is by passing something from one medium to another. So when we say one medium to another, medium just means like something from something like a glass versus water versus any other object. So the way lenses work is by refraction that happens because the light bends as it passes from one medium to another. The one medium here is just air, and when is in this water, which is another medium, you can clearly see that we see this straw bending. And this is not the straw bending, but it's what we are visualizing because the light is bending because of refraction. So that's exactly what is going on over here as well. And this creates an image on our retina that this object is much bigger, right? So once this light passes, it converges on and makes an image on the back of our retina. And this, to us, it feels like it's a much bigger object. So we can actually magnify it and see it by our naked eye. So the same principle, people used it for making slightly more complex microscopes. So instead of using just one lens over here, it's a combination of two lenses. One is referred to as an objective lens. So the objective lens is just the lens which is much closer to the object. So object here is an ant. The second one is called an eyepiece lens. That basically is closer to our eye, is going to magnify the focus of that object and will create an image on our eye. So now the light passes from this first lens, it creates an image over here. But since we have another lens over here, it passes through, and the apparent size of the image, so that's why we call it a virtual image that our eye sees now, is much, much bigger because now we are just magnifying it by two lenses. So often when we talk about the power of magnification is based on how much power each lens has, because more complex microscopes can also have multiple lens systems to keep on magnifying the system. But most of the microscopes work on two lens system where there is a one objective lens and there's a one eyepiece lens that helps to magnify the image. Now, the number of times it magnifies an object is really based on the power of these convex lenses. So let's say if this can sort of amplify the object 10 times and this can do it 20 times, you simply multiply it. So that means you're going to see the image 200 times bigger than what the object is going to be. So this is what exactly now we have in an encapsulated system for us to easily view it. I should have put some history about it, but one of the first earliest microscopes were by Leeuwenhoek, where he did see a bunch of different organisms, but also stuff including like his sperm, where he made this like basic tunnel microscope with different lenses and looked at stuff around him. So, okay, as we said, just the microscope is basically made of an objective lens that is used for the magnification. And usually this is the stage where we put the object. And then there is another lens, which you can't see here, but you are going to put your eye here and then view the whole thing here. So there's another lens here for more magnification. This, as I said, is the stage for the support. And these are just like the focus adjustment to make those knobs. So I know most of you don't have your foldscopes yet, but I wanted to just quickly show you how comparable a foldscope is to a classical dissection microscope in a lab. So you have exactly same system, what you see over here, except it's just made of paper and is very small. So here again, you have a lens here, which is your eyepiece lens. The objective lens for the magnification is on the back side, which we don't see. So this is really like an origami where you put multiple stuff together and build the scope. This is our focus. So this is something that moves, and Iqbal is going to show you in next classes how to use that. And then also has a stage on the back there where you put your slides and sample. So it's very, very comparable in terms of the components it has. Is very similar to a classical microscope. So just to give you a comparison of how much larger your image is going to appear. So a conventional dissection or an optical microscope can be anywhere from 40 times to 1000x. The foldscope has a limitation of 140x. However, foldscope can sometimes do even a little better, and we will go about it, but because nowadays most of our phones also have these beautiful, amazing lenses, right? And the one way to acquire images with foldscope is to stick it to your phone lenses. So you can also sort of exploit the lenses your phone has and the magnification power it has. So it's just going to keep on multiplying based on how powerful your foldscope is, but also how powerful your lens in the phone is to make those images. Okay, now coming back to thinking about algae in general. So what are algae? So one of the ways initially algae was thought about are these aquatic organisms that can conduct photosynthesis. But I kind of gave you an indication that algae can be so variable that it's very difficult to put them in one sort of classification, because algae can be eukaryotic also. So all the multicellular algae I was showing you, or also something like kelp, if you have seen in the sea, like these long filamentous plant-like things, they also algae, and they have the eukaryotic origin. However, some of the algae can be really prokaryotic as well. So again, when we think about prokaryotic, it's really how their DNA and everything is arranged. It will have a cell wall. The DNA will be open, will not have a proper nucleus and stuff. So, and most of these harmful algae, which we are going to talk about, like cyanobacteria and blue-green algae, are actually prokaryotic in nature. So it's very difficult to put algae in just like one category over there. Okay. So now we have been talking about that algae is broadly, we can classify it as an aquatic microorganism or organism that produces, that conducts photosynthesis. So what is photosynthesis? In a very simplistic way, we all know that photosynthesis is essentially what plants do, is like they take sunlight, take carbon dioxide from the environment and change it into sugar, and also take water and change it into some form of sugar, which is often referred to as glucose, and release oxygen. And that's the oxygen we breathe. That's why it's important to have plants around, right? But really to think about in a little bit of detail how this photosynthesis really happens in these plant cells. So if you think about any leaf that's made up of tons of these stacked cells together, again, as I said, these cell walls are really important to provide that rigidity. So if you see, this is like a beautiful brick, which is just sticking to next to each other. But if we go more and more detail in each plant cell, so this is one cell, that's another cell, that's another cell, that has another sort of organelle inside, which is called chloroplast. So each one of this is actually a chloroplast. But because people have been able to visualize more and more in detail, now we know that chloroplast is again a bilayered or bimembrane organelle. So there is an outer membrane, there's an inner membrane. But even inside that, there are these tons of small disc-like structures. So these discs are called thylakoids. And then the space between this disc and and this membrane is called stroma. So this is the empty space. And we'll go a little bit in detail why thylakoids and stroma area is important. So something that these chloroplast is full of is a pigment, which is chlorophyll. And can anybody tell me here that why do we see, I mean, it's written right over here, but anybody has an idea why we see plants as green in color? Anybody? Let me see, people. Why are plants green in color?
Because the red and blue light is absorbed, like it says in your slide.
Yes, that's why I said it's already written there. So, this is a very important concept as we will go further in classes, because especially it becomes important in spectroscopy, and Iqbal, I'm sure, is going to go into too much detail about it. But one of the reasons we see a particular color is the color which is reflected, not the color which is absorbed. So actually, when we say that the light is absorbed by these plants and is used for making food or whatever, it's actually not the green color, but it's the other primary colors, which is the blue and the red, that is being absorbed. So those wavelengths of light is being absorbed. And what is reflected is green. That's why we see. So green is kind of the useless wavelength for them. That's the wavelength they're not using. They're actually using the red and the blue. So that's always confusing when people say, oh, are we using a green wavelength for doing photosynthesis? No, that's the one which gets reflected. That's the one which we see. So all these thylakoids have tons of chlorophyll in them. So chlorophyll is here. Don't ask me the details of the structure. I'm not an organic chemist. So I always forget what all is here, but it's a pigment which basically is capable of reflecting green. Something I hope you can also see what is really striking about thylakoids here is how well they are stacked and how many of them are over there. And that's really to increase the surface area with all the chlorophyll, because each of these thylakoids inside will have small chlorophyll pigments inside. So if you can imagine that a plant has to do so much of, like, it has to make all that food, all that stuff, you basically want to have more and more surface area by actually not using too much of area. So what it does is it makes these like small disc-like structures to sort of have more surface area in a confined space. And inside there will be tons of these chlorophyll pigments. Okay, going to the next slide, just a little bit of detail about what really happens in photosynthesis. So photosynthesis is essentially a light-dependent reaction and a light-independent reaction. So it's actually two parts of it. So when we say a light-dependent reaction, it's very obvious that it needs light. Again, when we say it needs light, it actually absorbs the red and the blue wavelength. And what it does is it takes the water molecules, and it's kind of badly written here because it takes two water molecules and will convert it into oxygen, which is released because of this light-dependent reaction. And then it takes all the electrons, which is extra over here, after it breaks the H2O, And that is utilized by these intermediates. We don't have to remember that these are the intermediates, which are like the electron carriers. And these electron carriers now take the carbon, which is again coming in the form of carbon dioxide from the environment. And this is now a light independent reaction. So you actually don't need light. So there's already enough energy because of that electron release here that is taken by these intermediates that will take the carbon dioxide and fix it in a form of some sort of a carbon. And one way to form that sugar or carbon-based compounds is like glucose. But there are multiple intermediate states in between as well. So if you really want to go in detail, I will really suggest to look at this cycle over here, because the carbon fixation cycle itself is a huge process of its own. And then it's converted into whatever more complex sugars and stuff, and is kept in the plant. Now, the light-dependent reaction, as I said, happens in thylakoids, as this area uses the chlorophyll pigments here that does all the work. So actually, these sort of coin structure here is really important to create that sort of membrane potential and to carry electrons here. But the light-independent reaction actually happens in the stroma. So all that is now released in this area of the stroma. And that's where the fixation of the carbon will take place. So that's the light-independent part. Okay.
I just wanted to add a quick perspective. If you go to the previous slide, so these are essentially nature's solar panels. And the way the thylakoids are organized is to act like a battery. So each of the thylakoids are basically transmitting electrons and amplifying the electron passage to do the reaction that are later on will be helpful to create sugars. So it's already like well organized, and I found it quite fascinating that they look like batteries stacked up against each other.
That's true. Spoken like a true, engineer, which I am not.
But yeah, I mean, you can see that the efficiency will be amplified if they're stacked up like a battery.
Right.
Yeah.
Okay. So just to again go back to now thinking about algae. So algae actually is one of the, Iqbal, right? Like one of the oldest organisms developed on Earth. It's like 3.5 billion years ago.
Absolutely. Yeah.
And the biggest source of actually oxygen on Earth. So Earth is oxygenated because of algae. And we'll go to the fact that we actually need algae. It's not like we want to completely get rid of algae from Earth. Oh yeah, it's written over here, which I forgot already about my own slide, that the atmospheric oxygen concentration really skyrocketed because of this algae that developed on Earth, right? So it's also so diverse, like it has diverse functions. You have really a wide variety of algae you can find. And it's really important to maintain that aquatic biodiversity, because a lot of other organisms also depend on the oxygen that is produced by algae inside. So it's just not the oxygen that gets dissolved in the water, but it's also the algae which is produced in water itself. That is really important for the aquatic organisms. So it comes back to the idea that algae is really important, right? Sorry, there's something in the chat.
No, I addressed it. Don't worry.
Okay, sorry. So okay, so why do we want to care about what are the levels or what are the different kinds of algae? So this is a beautiful cartoon we see, because it's becoming, well, I say beautiful, but it's actually quite bad, because nowadays when we go to a lake or something in summer, it's often closed because of algae bloom. So clearly it's an issue as well. And that's where we have to start thinking about that algae can be good and bad at the same time. So what are algal blooms? So when people say that there is a blooming problem of algae, it's because of this rapid growth of some sort of harmful algae or microscopic algae that becomes like what people refer to as colored scum on the surface of water. So if you look at this Lake Erie in Ohio here, you can clearly see that you almost don't see the water there. There's this thin layer of algae on the top of it. It just looks green. And similarly here in Florida as well, it's this colored scum on the surface. And we'll go in a little bit detail why that is bad. This, I think we have a picture from the, what do you call it? This
Satellite.
I forgot. Satellite, thank you. The satellite had a brain freezer, the satellite image of algae, where you can see, you just not see the blue, but you start to see these really green kind of goo on the satellite image, which is not great. So again, like people over the years have noticed that this algal bloom has been increasing, not in just their fresh water, but also in the seawater as well. So there is a real problem. So again, as we discussed that there are tons of non-toxic algae or important algae that we need in water. And often when you look at those algae, it looks like either these rooted plants or can be also filamentous. And you can even have these like macroscopic, so you can actually see them with naked eye kind of algae. So these look like what you would think often that these are plants. Some of that might be plants, but a lot of them on especially freshwater or seawater is actually characterized as algae. However, one of the harmful algae which sort of evolved over the years is the cyanobacteria or the blue-green algae. So this is also called cyanohabs, which is the harmful algal blooms, and cyanobacteria is the kind of algae. So if you remember a bit what I was talking about in the back, the cyanobacteria is kind of a protist. So it's more of a archaic kind. It's less of a eukaryotic kind. And if you want to quickly sort of by your eye look at a lake or some water body and say whether this is harmful versus a good algae, you can kind of do it for some systems. So it might appear like a paint or a soup. It'll give some sort of a foul smell from the far as well. Like if your water body is really giving you some sort of a really bad foul smell, that means it has been stagnant. And if it looks green like this, or the algae is actually bubbling, that means it's a harmful algae. And you have to be very, very careful about either taking a swim there, taking your pets there, and stuff like that. I mean, there has been a lot of monitoring going on in a lot of places, but we also know that there are tons of water bodies that don't get monitored that often. And this also can be very dependent on the temperature, the weather. So we'll go a little bit in detail about that. So why are these toxic algae really increasing in the water system? Do we have somebody to blame? And it's always the humans. We have to blame ourselves for that. So what is going on is that because of the use of excessive fertilizers that people, because of agriculture or increase in the agriculture that kept releasing in the water, that acts as nutrients for a lot of these algae. And they use it as like, oh, yum, food. And then they just keep dividing. So if you provide it a right temperature and all the right nutrients, because you have to think about these fertilizers are really, really rich in that nutrients, which algae can potentially use in order to divide and multiply more. So also, you know, it's kind of always correlative that if you know that there has been some area or some water body where there has been release of these nutrients from the nearby agriculture area, there's a high probability that you will have some harmful algae growing on over there. So also, again, that climate change, as I said, is not helping, because if you have slightly warmer temperature, it gives them sort of a good, nice temperature to grow. Okay, so what are the harmful effects of cyanobacteria? And this is a very limited, you don't have to remember any of them, but this is like a limited one slide outlook of what is going on. So a lot of these harmful cyanobacteria or the harmful algae are really releasing neurotoxins. And especially these are very harmful for the pets. And I think some of them actually have some impact on us, but the long-term studies of such impact has not been done. It's still sort of a thing in process. If I look at the recent papers that keep coming and coming, like we don't know what like, let's say if somebody is pregnant and goes to that water body, drains these harmful toxins, all that kind of studies are not done, that what effects it will have on the fetus and stuff like that. So this is, I would still say, is a very ongoing area of research, because if I look at the papers, it's still coming up. But we already know quite some toxins like microcystins or anatoxins, and these are actually neurotoxins. So they actually attack your neurons and can cause some of the issues. So there was this, if you also look at the news, there was this news about 3 dogs dying in Spokane in 2021 by just because the dogs took a swim for 20 minutes in that lake. And not just for that, but it's also very harmful for the normal aquatic environment. So it kills the fish that is there, it kills the other organisms. If it's in the seawater, it also kills a lot of crustaceans and other organisms as well. So how is this happening, right? Like, because logically, if you think about it, algae is just supposed to be releasing oxygen. So the multiplication of algae should be really good in our thought process. Then how is this happening that it's being so harmful? So not just that it's releasing these harmful toxins, but what it can do is create this sort of a dead zone. So the way it works is that you have all these nutrients coming from either industry or from the agriculture that is released in the water that leads to the multiplication of these algae. It will release oxygen to a certain limit, but in that process, it'll go through its life cycle. So it'll also die at some point. So when algae die, and when that huge amount of algae die, it sinks to the bottom of the water body and starts to decompose. During the process of decomposition, it's reverse. It's actually not releasing oxygen anymore. It's actually using up the oxygen which is there in the water body. So that starts to create a dead zone. So now you have tons of algae. When you saw those images where there's like layers and layers of algae on the top of water, what it's doing is it's thriving and it's doing its life cycle. So not only it's dividing, but it's also dying. And every time that algae is dying, it's just sinking to the bottom and it's using up all the oxygen there. And that depletes the oxygen in the whole water body. And that leads to, again, dying of more organisms like fish or other living creatures over there. Now it is just, again, feeding onto the system, because once the other stuff also starts decomposing, it'll keep using more and more oxygen for that decomposition process. So that's what is the dead zone that these algae can really create. Iqbal, you can add if I forgot something.
Yeah, if I may add a few points here. So excess amount of anything is bad. That's something we all know. But since algae is at the base of the food pyramid, excess amount of algae can cause damage to the ecosystem. So if you can just measure how much algae you have in the water body, you get some estimate of what's the rest of the organismic ecosystem will look like and what's the status of that water body. That chain of thought can be easily put together and you can draw some conclusions. Additionally, too much algae on the surface of a water body can also block sunlight and oxygen coming in from the atmosphere and dissolving into the water body. So that's another byproduct or bad reaction that happens if you have too much algae in the system. So in this project, what we are trying to do is measure one parameter, which is algal bloom, and then trying to draw some conclusions on what the rest of the ecosystem would look like.
Yeah, thank you for adding that. That's important. I forgot about the sunlight thing. Okay, so as Iqbal kind of added, and I'm almost in the end of my presentation, I hope what you have realized through this whole thing is that there are two important things. One, as Iqbal already mentioned, is the amount of algal growth itself in the water body, which we are going to measure by spectroscopy, is basically sort of exploiting this idea that it absorbs certain wavelength and it reflects certain wavelength, and that's what the spectroscopy does. But I also hope that you appreciated the diversity algae has. The difficulty people over, or the taxonomists over the years have had to put it into one category because it's so diverse in nature. So what we want to do with Foldscope essentially is to look at that diversity you have it in your water body and make a conclusion based on that. Like, it's not a quantitative conclusion, it's a more qualitative conclusion to see what kind of algae diversity you find in a particular season in your water body. So let's say if you're seeing more the harmful ones, because we do have a list categorized based on looking at what kind of algae it is, whether it's a harmful one versus a good one, to know and to see if that changes when you're looking through various seasons. And that's where the whole scope becomes important. So not just because it's also fun to look at stuff, I would say always, especially for me because I'm a microscopist, but also to really characterize that, you know, whether we are seeing a good one versus bad one and if that changes across. Okay, so I think the next steps on the coming sessions is going to be some of the safety measures we always advise to follow while sampling water. Important thing would be the full scope demo and troubleshooting. We can also send you links because since you're going to start getting stuff, we will send you some links from YouTube about your full scope. Even though we will give you a demo, I would really encourage you to look through those videos because they are those fun videos where you can start already using your full scope and become comfortable with them by looking at small stuff, as I said, like pollen grains, anything you find like an onion peel. It's actually really fun and fascinating to do that. And then we will talk about the algal species classification. So we have a nice, what do you call it? Algal atlas kind of a thing, yeah, where we have everything characterized. And I think Sayak at some point will go through that algal atlas, and then we'll discuss the protocols and stuff.
Yeah, I mean,
okay, that's it. Yeah.
Claps for Ankita. I mean, that was a fantastic presentation. I think those of you who have already received the full scope, it may be a good idea to open it up and explore the equipment and ask us or email us any questions that you may have. We will try to troubleshoot with you during the next session. We are just waiting for everyone to get the equipment so that we all are on the same page, and then we'll start exploring the equipments in detail. And we are super excited to see what you all find in your water bodies, what kind of algae, as Ankita already mentioned. The broad idea of microscope is to explore all the different species that you find in your neighborhood. And the broad idea of the spectroscope is to do bulk assessment of how much of green stuff you have in your neighborhood or water bodies, basically, right? Because the spectroscope doesn't classify between organism to organism. It just gives you a bulk assessment of how much green stuff is in there, because it basically reads out how much light gets absorbed and how much light is transmitted, essentially. So both these equipments are essential and are considered the gold standard for estimating algal bloom. And we are trying to provide these gold standard equipment at a low cost, but without compromising on the quality of the assessment.
But something I wanted to add with the full scope, feel free to use it as much as possible. It's like, you don't have to fear about, unless you're like really twisting it and doing something, it's very hard to actually break it as such. And that's the good part of these frugal tools, because they're technically not so expensive, but also they're actually hard to destroy, I would say, because I use microscope which are $1 billion, and everything there is like too much for us, right? Like one thing which gets ruined, it's like, oh, it's really ruined and it's going to cost us.
Yeah. I mean, that's the beauty of these tools, is that you can break them and you can buy another one, or we'll try to give you another one. But the thing is that it takes some time to get a hang of any equipment that you use. So try to connect it with your cell phone, maybe watch a few YouTube videos on that. We have a bunch of interesting explanatory videos on our own repository, so that may be helpful to go through the videos.
I think Saya can send that in the next email. Like we'll send you a few links to go through.
Yeah, I mean, it takes some time to get a hang of the device. So, how do you connect a slide? How do you connect your cell phone to it? How do you focus? How do you take a good image? That will take some time. So these are the things we'll explore in the next few sessions. If you have any questions, please feel free to chip in.
Can I ask a question about the objectives of the study?
Sure.
I was thinking of working with a pond or small lake near where I live in Liverpool, which definitely has algal bloom problems in our new horribly hot summers. But I also wanted to look at the river, which I'm sat on the side of at the moment, which I've looked at in terms of its insect population, and there's lots of stonefly larvae, so it's very highly oxygenated. I guess blooms there are relatively unlikely, and I'd be studying it just because I'm interested in what's going on in the river. Does that lie outside of your study? Do you prefer places where bloom is at least a significant possibility?
No, we give you the freedom. I think the only thing we would be concerned about, and it's not really a concern, what we are going to ask or request SciEC is to give you two different. No, he doesn't need two different accounts. Actually, you can use the same
Not
account.
really, yeah. So just
Yeah,
as long as feel free to go to the river and the lake, that's perfectly fine. But at one point I'll create an account for you on our website. So when you upload the data, just make sure to mention which water body you took that data for, so that the data doesn't get mixed up. If we have a proper segregation there, you're mentioning this is the river and this is the lake, then it's perfectly fine.
So we'll have
we'll segregate it. I won't mix it up.
Yeah, we'll have 2 experiments recorded for you, so that won't be a problem.
Yeah.
I mean, we keep it very flexible. In the end, it's also, as we kind of told you guys in the last 2 it's just not about the environmental impact, but it's also sort of an educational part which we get excited about. So as many water bodies you want to do, it's really up to you. Do how many you want. We have no restrictions. Please explore. I think the fun part is actually exploring it really, and we'll make sense of data if it's really making sense. But at this point, we're not so much concerned about that.
Great.
Yeah, I have a question about testing for other things like pH, sodium content, other minerals, and so on. Is that, well, would that be a good idea? Do you have any recommendations about particular devices for that? I looked on Amazon, and the kits seem to go from about $2 to 50,000.
Yeah, I mean there are
Sorry, that's it.
Toolkits available online, let's say on Amazon, that you can buy off the shelves, that you can use to get an estimation of pH and also other nutrients or other things in the water, let's say lead and other chemicals.
But
that
you don't need an expensive
possible.
one. You just need that strip thing. Yeah.
In this project, we haven't shipped you or sent you a pH strip specifically, but we don't want to stop you from exploring the sample as much as you want. So if you are interested, I would say please go ahead and buy a pH strip. It's super inexpensive, and you can use it to get an estimation of the pH and report that. With some of the projects that we did previously, we actually asked our participants to take a look at the pH as well, and also monitor some of the other factors like smell that is coming out of the water bodies. So we didn't want to complicate this project too much. And
so the thing is, with the pH, I wanted to add that there is no direct correlation as such always. So it's hard to find a correlation between like at least what I haven't seen in studies. Of course, all these minerals and stuff affected. So it can be one parameter. So let's say if you have an inflow from an industry or from fertilizers and stuff like that, and if you try to use a more broad water testing strip, so it's also the water testing strips are usually not that expensive. Actually, we took some to India and we were testing just the normal water we get in the tap water there, and it was crazy the amount of minerals it has, which it shouldn't. Like some of them was going off the scale, which is not great. Sorry, go ahead, Ankit.
So if I remember correctly, so you also proposed that you wanted to look at a river where I think some dam is going to open and it will have some inflow of that dam water, right? So I think for that purposes, just having this observational data where you record the pH and the concentration of different minerals will be actually great. you are stuck on my screen, but yeah, I mean, we can discuss this over
They
email.
had initial opening project.
Okay.
Official opening.
Mm-hmm.
I mean, they've been, yeah, so it'll be very interesting. I'm happy just
We're losing you, Chip,
play
a
around and
bit.
try different things, I think.
Sorry. Yeah. One of our earlier participants I wanted to add also was thinking about looking
Sorry.
at temperature. Sorry, Chip, we lost you just like for a second, but I got the idea what you were saying. Okay, so yeah, so some of the participants, and it's not a compulsory thing, but I think what SciAC has nicely done, and you will see where you will upload your data, you can also add some additional parameters there if you want to, sort of ask more questions, because like one of the earlier participants were thinking about the temperature, which is also a good thing to think about, like with how much is the temperature in those, like an average of what the end sort of weather looks like in that particular month or something, is also an interesting parameter to think about. Like if, like the David was saying, like you have hotter summers now, and that average temperature overall can really impact blooming as well. So that's another thing you can potentially look at. And that's something you don't have to actively measure, because nowadays it's great. You just Google it kind of and get an estimate of what the temperature was that day. Any other questions?
What are the I'm sorry, you said this, but I missed it.
Mm-hmm.
What are the cyanobacteria are prokaryotic? What other algae families, classes, whatever, would you regard as prokaryotic?
Iqbal, do you know the list? Is chlorella also one of them, right?
Yeah.
You're muted. Iqbal is better in that characterization than I am. But I remember chlorella, and there are other blue-green algae as well, which are in the protist family.
I think chlorella is eukaryote.
Is it?
So
I don't
yeah,
know. Just on a basic Googling.
yeah. So I think
Maybe not.
most of the algae that you will be able to visualize with the Foldscope are going to be multicellular, but some of them are single. I mean, the most abundant ones are also single cell because they're dispersed. The green soupy thing that you see is basically single celled organism algae, mostly belong to the family of chlorella, and they're dispersed. So you can concentrate them and visualize with your Foldscope. But the blue-green algae is called cyanobacteria, but it doesn't look like a traditional bacteria. Traditional bacteria would be really hard to visualize in a Foldscope because it is submicron, right? So that means that it is on the order of 1 micron or less.
But most of the cyanobacteria don't have a well-defined nucleus. So it's still kind of in a older archaic family than a eukaryote.
But the good thing is that sometimes they form colonies, and sometimes they are filamentous, right? They stick to one another like Lego blocks, and they make a filament. So that you can visualize. And the other toxin-producing algae also form filament, and they can move around like Oscillatoria. They don't have legs and hands, but they can move around. But to answer your question about whether they are eukaryotes or prokaryotes, I think we'll have to dig a little deeper, because we don't have a clear, I mean, we have a clear classification, but it's not right on top of my head. Like exactly, does it have a nucleus or not? We do focus more on what's the organization of the chloroplast there. Some of them have dumbbell-shaped chloroplast, some of them have spiral chloroplast, and so on. But
I wanted to broadly add that cyanobacteria mostly so the way that taxonomists also characterize things is a little confusing because everybody has different way of classification. But the name itself was given cyanobacteria because of not well-defined nucleus, which is one of the important features of it to become a eukaryotic. So I'll technically be protist, but again, there are going to be taxonomists which fight about it as well, that it's not really because of different regions, because it has a beautiful chloroplast in it, seems more evolved. So it's somewhere kind of in between, and it doesn't have a very rigid classification like that.
Yeah. Sounds like the challenge of trying to define species. It's
always.
reality is a bit more complicated than classification.
Yeah,
One of the biggest
nature,
fear of my life is taxonomy.
yeah. One of the things that we see quite often is called oscillatoria. And I'm just taking a look at it. It doesn't have a nucleus clearly. Yeah. So it is.
So some of the algae are also very interesting because what they do is actually initial stages, they have a nucleus and then they like squeeze it out to make space and have more chloroplast in it. And that's kind of interesting too, because there will be only few of them when they're stacked together that will have a nucleus, and some of them will then in between will be empty. So it's just like smart way of making, like having more and more chloroplast and then just defining in that organization that some of the cells are just going to have the nucleus while you don't need that in all of them.
Yeah, nature is quite diverse to actually fit into one category. Seriously.
But that's an interesting question and also something to think about. Yeah. I'm sure somebody will see an oscillatoria. It's like one of the most common ones we see. And any other questions?
Can I ask a quick question?
Yeah.
So do these algae compete? So in a pond or a lake, can there be competition between algae?
Yeah, I think so. They always compete for food, which is primarily sunlight and oxygen in this case. So proliferation of, I think it would be safe to say that proliferation of one species always causes a problem for other species. So yeah, if let's say chlorella there's an overabundance of chlorella, then it blocks the sunlight and it will consume more oxygen, and that will cause problem with other algae. But yeah, I don't think there's any evidence that they work.
Exactly.
There could be symbiotic relationships as well. I'm not really aware of it.
Yeah, that was my question. Okay.
So I know that there are ecological competitions between algal species as well. And again, it depends on the rate of division and the kind of nutrients particular algae need to divide, because the rate of division for different kinds of algae is also very different. So you can essentially think about like if there is something which divides faster, so it's almost like cancer cells with the neighboring normal cells, right? Like tumor cells, because they have such a high power of division, eventually tends to win more and more unless you have a mechanism to stop it. And that's pretty much is what ecological competition in system like this is also. So yeah, there are other factors also so there's definitely one can win. Yeah, I'm sure there's a net logo sort of something done somewhere about it. Sorry, we just go off track often in our own.
I think we've already gone over the 1 hour time limit.
We will release you if you don't have questions.
Yeah, hopefully in a couple of weeks, everybody will have their equipment and will be in a better position.
But I really want to thank you guys for being such an enthusiastic group. It's almost more fun for us to learn as well, because we learn in the process as well. Okay, we're good to go.
Okay, yeah.
Happy weekend.
Thank you, everyone.
Happy weekend,
Yeah,
happy
thank you.
Easter, everything.
Bye.
Bye.